Hello everyone. In this video, we will be talking about ATP as the title here that we have would suggest. All right, so starting off, what is ATP? ATP is a high energy molecule, and if we break up the name ATP, it's adenosine triphosphate. So right off the bat, we can derive some information from the name about this molecule, and that is that it has a molecule of adenosine in it, as well as 3 phosphate molecules, and these phosphate molecules are actually phosphoric acid, and if you remember from a previous video, the structure for phosphoric acid is right here. So we'll get into this in just a second. So we want to go over some basic general information that's good to know regarding ATP. So starting off, whenever a molecule of ATP is hydrolyzed, it releases 7.3 kilocalories of energy, and you can convert that to joules if you want. Like I said, it's built out of 3 parts, and we'll go ahead and list those parts. It is usually, when you look at it, you first notice the nitrogenous base. After that, you have a ribose sugar, I should say, and then you have the 3 phosphates. And we'll go into the molecule here in a second. After that, we need to know that ATPase, that's what breaks down ATP. And if you remember enzymes, ACE breaks things down. So on the other side of the spectrum, ATP synthase would do the opposite, because this is synthesizing is to build something, so a synthase is going to create ATP, whereas just a regular enzyme with the suffix ase is going to break something down. So these 2 enzymes are fundamental when we're talking about how to, well, one thing, conserve energy and also release energy with ATP. So I'm going to go ahead and scroll down here. And what we're going to do next is kind of construct a molecule of ATP, starting off with the basics. So, and what I mean by that is I'm going to write draw. And if you guys can do this on your own, go ahead and try, and then come back and see if you were close to what the actual structure looks like. So basically, we're going to draw a nitrogenous base, and it's going to have to be adenine for ATP. So we're going to draw the nitrogenous base adenine. Then we're going to add on to it to make it a nucleoside. Then we're going to make it a nucleotide. And then lastly, we're going to turn it into ATP. So if you can remember the steps of going from one of these to another, you'll be able to construct ATP just starting off with the nitrogenous base adenine. So to start off with adenine, I'm going to make it over here because ATP is a long molecule. We're going to start off with, it has quite a few nitrogens in it. So you're going to start off with a ring with nitrogens in it. And it's actually not super complex to remember it. If you think about it, it's kind of just a benzene ring with a few nitrogens in it. And if you just remember that your 2 double bonds on the right side of the molecule both have ends attached to them, then you basically have half of your adenine molecule completed. So then moving on to the next part of our little structure, and this part's similarly not too complex in nature. And so right here, this is already adenine. We already have our nitrogenous base. So this is, I would say 1/4, but drawing ATP after you have the nucleotide is really easy. So this is already like 1/3 of our entire structure drawn already. So now that we have our nitrogenous base adenine, and I'll switch the color here just so we can show each part. Now we're going to draw our part that's going to turn this into a nucleoside. So you want to draw a connection here, and then we're going to make that into a little carbon segue. And this probably won't be the greatest cyclohexene you've ever seen. Well, it's not even going to be a cyclohexene because we have an oxygen here. And this is going to be our ribose sugar. And if you can tell, because that oxygen's there up top here, if this was, once we draw the nucleotide here, this would be a constituent of DNA because of this oxygen here, because DNA is, or I'm sorry, not DNA, it'd be RNA because RNA has the oxygen and DNA doesn't because it's a deoxyribose. And then we also have a small OH coming off right here. So now we have our nitrogenous base and we have our nucleoside. So this whole structure is the nucleoside, and this whole thing is what we call adenosine. So now we want to turn this adenosine into a nucleotide. And the way we're going to do this is, it's a little complicated to show on this whiteboard, but all we're going to do is add a phosphate group, phosphoric acid. I'll go ahead and draw OH, OH, and then so we have OH, but we need to connect these two segments together. And the way we're going to do that is through dehydration synthesis. So what's going to happen is, to bridge these two, we're going to have H2O come out of this. And so what's going to happen is that this H is going to disappear, this H is going to disappear, and this oxygen is going to disappear. And what we're going to end up having is well, we can just bridge the molecule right here. And then I'll go ahead and erase this hydrogen and this OH. So as you can see here, now we just have a straight segue between the two, connecting the oxygen to this entire structure. So moving onwards, now we have our nucleotide, and our nucleotide is AMP, adenosine monophosphate, because there's only one phosphate, and we know how to connect these two together. So now we want to add on 2 more phosphate groups to construct ATP. And so the way we're going to do this is pretty much the exact same thing that we did over here using dehydration synthesis. And I'll go ahead and erase this hydrogen to show that. So we're going to erase the hydrogen. And then, because let me draw this structure of phosphoric acid again for you guys so I can show this a little better. Except for the double bonded oxygen, every other oxygen has a hydrogen attached to it as well. So if we were thinking, how would we attach and actually this was, I don't think I drew the H here, but no, I did, I erased it, didn't I? Okay, so we erase that H, and that means we have to get rid of this H and this oxygen. So the way we're going to do that is by the dehydration synthesis, where you lose 2 H's and 1 O. So like I said, we're going to lose this O, this H, and we already lost this H. So that means we're going to have this structure right here. And this is going to be ADP, adenosine diphosphate. And so we want to do the exact same thing we just did. We want to do another dehydration synthesis reaction to attach another phosphate group onto this. And so let's just go ahead and do that. And now we have ATP. So this is our entire ATP structure, and we constructed it just starting with our nitrogenous base adenine. So after you draw this a couple times, it's really not too difficult to draw it after you've done it 2 or 3 times. You kind of get a feel for it. But now that we've done that, let's go ahead and identify the functional groups that are present in this molecule. So looking at this, I think that the most easiest one to identify right off the bat is this alcohol group here, and there's one right next to it as well. And I'll go ahead and circle it just for the sake of it. So we have 2 alcohol groups here that are pretty well, I mean, we have a lot of alcohol groups, but these are the only 2 attached to the carbon. So we have a bunch of alcohols, and this one for me is another easy one to identify. I always look for the oxygens because oxygens are very apparent within molecules. So any oxygen that's between 2 carbons is going to be an ether. So we have an alcohol, an ether, and then after oxygens, I think it's easy to look at our nitrogens, and we see here that this nitrogen has a double bond. That means that this nitrogen is an imine. And similarly, if we look at this nitrogen, there's no double bonds, only single bonds. That means that this is an amine. And looking at this, since it's attached to no hydrogens, that means that this is a tertiary amine. And the way you remember that is so we have a NH2 over here. So this is going to be a primary amine. So you can kind of see the difference here. A primary amine will only have 1 R group, and a tertiary amine will have 3 R groups and no H's. So if we had a nitrogen that was bonded to only 1 H, then that would have been a secondary amine, but I don't think there are any in this molecule. Next, we can just look at this simple double bond between 2 carbons. This is a alkene. And then I believe that there is only 1 more, and that's going to be our phosphoric anhydride, which is going to be pretty much like this entire molecule right here, this entire group. This is a phosphoric anhydride. And that should cover all of the functional groups within this. I'll go ahead and circle this. If you guys see any more, feel free to let me know. But we're going to go ahead and move on from there, and I'll just go ahead and point out that this is a secondary alcohol down here. But yeah, like I said, moving on, we are going to briefly talk about how ATP and ADP is, how it reacts in the mitochondria. So we'll go ahead and write mitochondria right here. All right, so mitochondria. So if we're trying to synthesize ATP, this is a useful reaction to know. It just kind of helps solidify our understanding of how ATP in general works. So to synthesize ATP, we want to start out with ADP. And we know that we have to add on a phosphate group to that. And if you remember from a previous video, I said that inorganic phosphate is synonymous with the phosphoric acid that we use with ATP. So we use these 2 together, and we have to supply the reaction with energy. And so the same amount of energy that ATP releases has to be put back in to reconstruct the ATP molecule. That's why we have to eat food. So ADP plus inorganic phosphate plus 7.3 kilocalories of energy. And when they react with our ATP synthase, this gives off ATP. And it should be noted that the ATP synthase is inside the mitochondria, and it's kind of what bridges the gap between the inner membrane and the matrix, the mitochondrial matrix. And we'll get into that in the electron transport chain in a much later video, but this is a very basic understanding. And we can just reverse this reaction and do ATP, and it'll undergo the ATPase, the enzyme, and then it'll give us ADP plus inorganic phosphate plus 7.3 kilocalories of energy. So this is kind of what our reaction looks like that takes place inside of the mitochondria. And just one more thing to note with ATP, and this is just one of those random things that might pop up, you never know, is that ATP can cause, so let me rephrase this. We know that ATP requires H2O in these reactions. H2O is what either synthesizes the reaction or breaks it up through hydrolysis. So when we have our, and I'll just draw two phosphates right here next to each other. We know that this is a part of our molecule, and this is really what's going to happen when the molecule breaks off of phosphate to release energy, is that this H2O comes in to this bond, and one of the hydrogens is going to attach to that middle oxygen. So then we'll have one phosphate group here, and then OH is going to attach to this one. So we end up getting identical inorganic phosphate molecules over here. And actually, if I think about it, this is going to be attached to the rest of our ADP molecule now. However, now that we have this broken apart, we see that H2O is required to release the energy that occurs with the phosphate groups within ATP. So you can imagine what happens if we have a lack of ATP, or not ATP, H2O, and we are dehydrated. If we are dehydrated and we don't have a significant amount of H2O in our system, this entire reaction can sometimes come to a halt and can cause significant problems. And that's the reason that people sometimes faint when they're dehydrated or have other medical issues due to dehydration. It just kind of goes to show you how many different elements are at play with something, as simple as H2O. H2O is probably one of the most important molecules in the body. It's what are we, 73% made of water? That's where 73% of our weight comes from. So that kind of just goes to show you how important all of these different elements come into play and how they work together to create life for us. That being said, that's going to be the end of the video, and if you guys have any questions, feel free to let me know.